Hello everyone, my name is Roselia Ayala-osorio and I am a second-year master's student in the Aquatic Sensory Ecology Lab at the University of Windsor. And today I will be presenting my research on the integrative response of the round goby to multiple stimuli. Fish are surrounded by multiple stimuli within their habitats, which provides important information about the complexity of their environment and what type of abiotic and biotic factors are present. This could be anything from predators, prey, conspecifics, or ships or boats. The ability for them to perceive this information and respond to signals can be an important benefit to their survival and allow for fish to communicate with each other. Fish can communicate through visual, acoustic, and olfactory signals, as well as electric communication and mechanosensation, which help them navigate and sense their environment, but we will be focusing on the first three as it pertains to my research. Visual signaling can involve behaviors such as gill flaring or physical courtship. Fish can also produce sounds and perceive sounds. Some example of these vocalizations can be grinding the pharyngeal teeth, or the use of the swim bladder, where it acts as a drum when it's vibrated by the surrounding sonic muscles. Teleos fish produce vocalizations that are important in mating decisions and agonistic settings. Olfactory signals can also help in sensing pheromones of potential mates, species recognition, alarm, and species aggregation. Previous research has used a variety of stimuli on fish to elicit behavioral responses with the aim of understanding how animals behave in their natural environments. Visual stimuli can include presentation of mirrors, conspecifics, or video playback. Acoustic stimuli can include mating calls or different types of anthropogenic noise. And olfactory stimuli can be the use of pheromones to study mate attraction. Previous studies have focused on the use of single or coupled sensory modalities, so it's important to move towards integrating multiple stimuli on fish so we can further emulate their natural environments. Therefore, the objectives of my research is to determine if reproductive male round gobies can exhibit an integrated or synergistic response when presented with visual, acoustic, and olfactory stimuli. To further define synergistic response, this simply means that the intensity of the response will be greater when presented with three stimuli simultaneously than the sum of responses to individual stimuli. My second objective is to determine whether a relationship exists between the intensity of the behavioral response and sexual maturity. My study species is the round goby, and the round goby is native to the Black and Caspian Seas. It was introduced to the Great Lakes in the 1990s by the ballast water of ships originating from its native area. It has become very invasive in the Great Lakes and outcompetes many native benthic fish due to its ability to adapt to a wide range of environments, has a broad diet, and spawns repeatedly throughout the spring and summer. Reproductive males of this species turn black in color and have puffy cheeks. When males are ready to spawn, they create nests, and from the nests they produce calls, visual displays, and release a sex pheromone to attract potential mates. The urogenital papilla can also be assessed when determining reproductive status. The papilla becomes swollen, and this is also the case for females, where the only difference is that it becomes swollen and orange in color. Fish were caught via angling at two locations within Riverdance Park in LaSalle, Ontario, then brought back to the University of Windsor to conduct experiments. My experimental setup included an underwater speaker to play the acoustic stimuli, which was a male goby call played at 150 decibels underwater. This falls within the range of the round goby hearing and does not harm the fish. I used an iPad to play the visual stimuli, which was a video of a reproductive male round goby exhibiting resting and swimming behavior. And for my olfactory stimuli, I used a liter of conditioned water using a reproductive female. So I created this by placing a reproductive female in 1 liter of dechlorinated water for 4 hours, and that was used as the olfactory stimulus. Following experiments, we took the body measurements of each fish, which included total length, head width, body weight, and gonad weight to calculate GSI. Each experiment began with a 30-minute acclimation time. The experiments alternated between 5 minutes of control and 5 minutes of the chosen stimuli, and finally ending the experiment with a presentation of all 3 stimuli. Each control consisted of a period of no sound, no visual stimulus, or olfactory stimulus. The following behaviors are what we quantified in terms of the time spent exhibiting these behaviors. The first behavior is an approach, and this was defined as a fish traveling in a straight line towards a stimulus, and the distance traveled needed to be at least 1 body length. The next behavior is swimming. So swimming was defined as either turns, short swim distance that were less than a body length, swimming in other directions that were not towards any specific stimulus. We also quantified time spent resting as well as respiration rate, and finally erratic swimming where fish would move up and down the water column. For the sake of time, for today's presentation, I will just focus on the time spent swimming analysis. Our results show that there was a significant effect of the olfactory stimulus on time spent swimming. On the graph, you'll see that the zero line in bold indicates a baseline where the stimuli had no effect on swim activity and is no different from control. Therefore, if we look at the acoustic stimuli, you see it's very close to the zero line and likely had no effect on swim activity. However, the visual stimuli caused a reduction in time spent swimming, and we also see this same reduction in time spent swimming when we present all 3 stimuli simultaneously. Our GSI calculations showed that the majority of the fish used for experiments were in fact not really that reproductive, despite showing external characteristics of sexual maturity, with the exception of 4 individuals that really had high sexual maturity. So what do our results mean? In terms of the acoustic stimulus, we don't really see an effect on swim activity since it's so close to that zero line. Due to the fact that the majority of the fish were mainly non-reproductive, it doesn't feel threatened by hearing the call of another male and therefore does not really need to defend their territory. The visual stimulus showed a decrease in swim activity and could be explained again by the fact that there was low sexual maturity among many of the fish used for experiments and therefore didn't feel threatened by the presence of another male. In contrast, we see that the olfactory stimulus shows an increase in swim activity. This could potentially be explained by the priming effect by using pheromones to prime fish to influence them to behave in a certain way. We still need to analyze this a bit further to see exactly what is happening at this point during the experiment. And finally, we have the effect of all stimuli presented simultaneously, which cause a decrease in the time spent swimming. And this could be because we have the visual component of the experiment also incorporated at the end of the trial, despite having the olfactory stimulus present as well. It could be that the visual aspect kind of overrides that and still causes a decrease in the average time spent swimming. The next steps in my research are to examine the changes on the other quantified behaviors and to further determine if a synergistic response is occurring. I hope that my research can shed light on the integrative responses in freshwater fish and can be a stepping stone to determine if multimodal signaling is worth the energetic expense and if its use is a good indicator of fitness.